found that the critical point of this regulation was occurring was by RNAs from going from transcription to translation. And this RNAs was targeting transcripts that were important for anaerobic physiology, but the RNA itself was regulated in the oxidative physiology. Okay? So the question is, why would the cell do that? Why would it use a transcription factor? So we did some modeling. And it turns out when the RNA is doing this, it responds much faster to environmental changes than a transcription factor. So the graph on the left shows that the response of this circuit to changes in oxygen is much faster than the graph on the right, which is the response of a transcription factor. So the model predicted that if we remove the RNAs, the functions are still there, just they're made at the wrong time, they're not coordinated. And because the pump is maybe made at the wrong time, it's going to suck up all the ATP, the ATP is going to go down. The fitness of the cells is also going to drop. And these are testable hypotheses. So we did remove the RNAs, and we saw that the ATP levels just crashed. And depending on RNA composition, there was severe cost of fitness. Okay. So this is a point I want to make or emphasize. This RNA is seed in humans, in rats, you see in other organisms. It doesn't deal with solidity. It's how it's wired into the network that gives it its special property. Who is correlated with, who is targeting, and which environmental context it's responding to determines how the circuit is operating. So here we have figured out how this archaea is dealing with high salt, and it turns out to be the wiring diagram mediated through an RNA. So this is just one, I'll give you one example, there's many such examples, but as I said, it's a 5.8 trillion light year high stack. We went after one, and just from that one, we got all these insights about how these are connected to each other, how networks evolve, how organisms adapt to new environments, how gene family expansions drive niche adaptation, and some very fundamental insights. You don't need to work with E. coli and yeast to get fundamental insights. You can do it with any of the microbes, other microbes. So that's career advice is typically the NIH, NSF, and the funding. I don't fully understand the funding environment here, but the study sessions are biased towards people who look at mainstream systems. And there's typically a undertone of skepticism when you look at something that's out of the ordinary. And I would argue that for most of the systems that people have looked at for decades, you are reaching an asymptotic plateau. It's time to look at new organisms. Now, what ties the biology of all organisms is how information is encoded in DNA. So we look at one aspect of environmental response, but the tools, the approaches, the ideas developed were readily applicable to infectious disease and to complex genetic diseases as well. So I'll give you two examples. One has to do with cancer. And we took patient data. We took about 270 patients with different types of glioblastoma or gliomas. And then we asked, can we use their transcriptional profile to understand the disease better, to see what are the signatures of the disease progression. We'll apply the same algorithms, but one thing to keep in mind is biology varies from one system to another. So you have to really respect the differences in mechanisms and the evolutionary basis for these differences, because that really tells you how you can change the algorithms to extract the right kind of information. So we applied this to making the right tweaks, and we found groups of genes, found many clusters. So there were these signature profiles that we were able to extract. These are genes that are changing in a coherent manner across patients. And you can then take the clinical diagnosis of the patients post-op and ask, how do these trajectories predict and explain the clinical outcomes? So here you see that when the expression levels of genes are low, the patients predominantly have a normal diagnosis. And as the expression levels go up, you go into astrocytoma and eventually into glioblastoma. And there are a set of patients where this signature just does not exist. So simultaneously we stratify disease and stratify patients. And by doing that, we can also link to the specific mechanisms that are responsible for the signatures in these patients. And we developed algorithms to then find from these signatures that are found in the 3' untranslated regions, microRNA binding sites in these transcripts. And by building a hidden knockoff model, we pulled from a database of microRNAs in humans a specific microRNA that was responsible for the signature. And we did this for 46 different cancers. And that gave us this network, which essentially answers to the subtypes to specific microRNAs that were responsible for driving the signatures in those subtypes. So essentially, we've taken cancers, found the microRNAs that are driving the observed trajectories in those cancers, specifically related to these signatures. And because the signatures eventually tie into genes, the genes can allow you to then deconvolute the information regarding the biological processes affected. And so here we can now design experiments where we can say if we perturb this microRNA in a particular way, the expression of these genes will be affected and this phenotype will be affected. And so we did that. I won't go into too much detail, but we're tuning the expression of microRNAs occurring in mimics, and we show that the genes that were predicted to be controlled by microRNAs are responding in an equivalent manner. So we did the same for TB. As you know, there's about 1.5 million deaths caused by TB each year, about 8 to 9 million cases. Many of these cases end up going into latency. And the question is, can we figure out what the networks are that are associated with latency TB? What are the networks that get activated when TB comes out of latency? Can we trick the organism to show itself when it is not expecting an attack, and then we can drug it? Right? So we built a TB network in collaboration with David Sherman at Seattle Biomed. And in this network, we can then look at known drug targets, and we begin to see very interesting interactions among genes and drugs. And that gives you some ideas about possible combinatorial strategies that we can use for drug repurposing and combinatorial drug therapy, also in a patient-specific manner. So there's many different applications right now that are underway. With climate change, we just published a paper in PNAS where we've taken a diatom and asked how does it respond to climate change. For bioremediation, we're looking at reducing organisms. Biofuels, we're looking at methanogens and also algae, different kinds of algae. We're working with a company called Sapphire down in San Diego, who by far are the most progressive in fuel production. And they have increased fuel production by a factor of 2. And they need to find the critical genes that they need to perturb to get to that level so that they can be competitive with fossil fuels. I mentioned with TB and cancer as well. And with that, I'll stop. And I think I have 5 minutes to take questions. Thank you.
Questions for Nitin? Let me just start with a real high-level question. So you're studying very simple organisms that cover very complex processes. So in looking, so people that are studying humans are studying much more complex organisms. Are the same rules applicable? I mean, or is the complexity when you get to that multicellular level and you get to that organismal level, does it just complicate things so much? I mean, what makes you stay with a much simpler organism? And what's your drive to do that?
Well, 2 reasons. One is Australian microbiologist, so I understand microbes better. But I've started looking at complex systems, and I think the approaches don't change. It's the tools and the tractability of systems that changes tremendously. And working with human systems is the hardest because you don't have control over the populations. You have to have cell lines which don't completely model the human system, and in the mouse models, which do that even poorly. But I think there has to be an integrated approach. And the way we're doing it is by starting with patient data and trying to figure out what's most relevant to patients, and then looking at what is modeled with regard to the things we discovered in cell lines and in the mouse models. So we can do the experiments to test them on the hypothesis. In the end, all of these bioscientific approaches will give you predictions, but 80% of the time it's just slightly off. And you have to go back and test them. And that's why the microbes are the greatest, is because you can create these large libraries of things very quickly. Mouse is also very good at that, but going back and forth between mouse and human systems is one way, I think, at least we're thinking of overcoming that challenge.
So let me just ask you another question, if there isn't one. So it kind of gets to my next question, which is really drilling down there. If you look at the validity of the models that you're developing, and you look at your colleagues at ISD that are doing this in more complex organisms, and when they go to validate, what are you learning there? I mean,
exploring any other really developing technologies to see if we can look at signals and responses from the system. So there's a component of technology development that becomes essential. So this, you can go to a certain extent with public resources, but we taper off very quickly. So one component of ISP is our mantra is biology-based technology. And with D as a president, I think that's in our DNA to identify what really needs to be measured. And if the tools don't exist, we have to develop it ourselves. And then develop the computational tools to analyze new kinds of data. And then eventually we have to go back and test. So I think that's the way you work with the challenge.
Questions? Yes.
This is back to the network function. And then you also talked about some unsupervised clustering, right?
Semi-supervised, yeah.
So how do you incorporate this?
The semi-supervised cluster?
What is the example?
Oh, so there are two steps. So the supervised clustering is the precursor to the ensemble modeling. So what we do is we take all known information regarding gene functional associations as prior knowledge. And then we say, there's the expression data and other types of information. We'll start using some of the experimental data, bring in the clustering. So we find 4 genes that are somewhat correlated. And then we look to see which other genes can we bring in to test whether it's correlated to these 4 genes. And then we use that framework that we have on all the functional associations to prioritize the genes that need to be tested. And when those genes come in, we can do cross-validation against expression, we can cross-validate against nucleotide level mechanisms, and other kinds of independent sources of information, and select, keep or reject the views. And so that gives you the clusters. And the ensemble comes in when you have a very vast data set. If you try to do this approach over the entire data set, you tend to go into this major attractors, which is the dominant stakes. And you don't want to do that. So what you do is you take subsets of experiments and you do your ensemble clustering, sum on each subset, and then you combine everything. So you can mine the entire space as deeply as possible.
Other questions? Don?
So I'm interested, I mean, the technology is greatly improved, you're applying it to signals all over the business. But as you go forward, presumably you'll be able to apply in a larger sense, but what's the rate limiting step you're currently encountering? I mean, it's not sequencing, is it on the informatics side, or where is it?
No, the rate limiting step is the validations. So the way we can generate data is, as you know, is high throughput. Everything people does its own genes nowadays. And you can do it over many environments. It's automated, you have multiplex technologies and so forth. Algorithms computing is progressing very fast. You can do parallel computing, high performance computing. So all of that tends to go very fast. And then you get these networks. The networks give you hundreds, thousands, or more predictions. And each of these predictions require testing with different assays, different types of mutations, and so forth. That becomes very expensive, and it's a real rate limiting step. And in the end, you choose 1 or 2 things because you're publishing data. So I think we have to really think carefully about how we can get the outcomes. And that's why the drug screening platforms that have developed could be a great way to do likelihood testing. Because if we can see how these networks correlate with drug responses, because drugs have targets throughout the genome. You can use drugs as perturbers, so you can then block or change the network to test this also. And that connection is, people are just beginning to think about it in clinical genomics also.
Can I ask another question? Hello. So your challenge, if I understand, is not producing data. We can generate a great number of data to the bioinformatics and the pathways, but then validate it. How much we have used other omics technologies and the integration of different omics technologies to narrow down those final questions to be validated or targets to be validated? Integration of genomics, proteomics, metabolomics, and to narrow down results.
The inference should integrate a lot of data, so the first step itself of analysis should have an integrated component, because these data sets tend to, because they're so vast, they also have a significant amount of noise. And to get with the